Hi everyone, I'm happy to be here today to be talking about the correlation between cortical morphology and thickness due to forces generated during folding. And I want to acknowledge from the start that most of this work was done by my graduate student Nonhuman. Cortical thickness is a biomarker of neurological health. Abnormalities in this thickness are associated with a number of mental and neurological disorders, including autism spectrum disorder, epilepsy, and schizophrenia. And when most people talk about cortical thickness, they're talking about a single scalar value, which is the cortical thickness at a bunch of different points averaged throughout the brain. But in reality, the cortical thickness varies throughout the brain. If we look closer and observe patterns, we'll see that these outward folds, sort of the mountain peaks, which are called gyri or singular gyrus, tend to be thicker than the sulci or singular sulcus, which are these inward folding valleys. So it's been known for a long time, for over 100 years, that gyri tend to be thicker than sulci. But this sort of leads to the question of why or how. So in some work at the end of my PhD, we showed that cortical folding naturally leads to thickness variations in a sufficiently soft film and substrate that starts out with a uniform thickness, growing uniformly. When buckling starts here at the top, the gyral sulcal thickness ratio, so the ratio of the thickness between those outer and inner points, is very close to 1. But as buckling progresses, we see that ratio increase until it approaches actually even 2, so where gyri would be twice as thick as sulci. We showed a similar trend in stretched polymer experiments. And I want to acknowledge here the co-authors on this work, who were my PhD advisor, Ellen Kuhl, and 2 collaborators, Alain Gorielli and Sylvie Boudet. So after that work in my independent lab, we've been exploring this issue further. And a question that has come up are about tangential folds. So what I'm talking about here is not this gyri-sulci binary. These gyral ridges, like a mountain range, are not linear along the surface. They might bend and curve, intersect and split. And so I've highlighted a few representative tangential folds on the surface of the brain. So if we believe that the forces of folding in this direction are causing thickness changes, then it stands to reason that folding in this plane could also affect the thickness. So I posed this question to my graduate student, Nonhuman, and she suggested using the shape index, which is a single scalar quantity that indicates the shape of a local point. So it assigns a scalar value between -1 and 1. And so it sort of gives us this spectrum from the most concave shapes to the most convex shapes. So when we look at the human brain and characterize its surface in terms of shape index, we see that there is a variety of these different values along the surface. And the interesting thing is if we divide this shape index up into the 9 dominant shapes, and look at the cortical thickness distribution in each of those shapes, which is shown here in these different colors, we see a smooth spectrum of cortical thickness from the thinnest, most concave shapes to the thickest, most convex shapes, and with other shapes in the middle. So what this suggests is that it's not, again, only a gyral sulcal binary, where a point is either gyral and thick or it's sulcal and thin, but there are sort of points that are more or less gyral or sulcal, more or less concave or convex, and that affects their thickness. So after that work, which was published earlier this year in Human Brain Mapping, we've been interested in also thinking about non-humans. How generalizable are these observations? Is this phenomenon? And do we see it in other species? So we're first looking at primates, and here are some images of the primate species that we have looked at. And the great thing about primates is that they really offer us a rich sandbox for hypothesis testing. So we have a huge range of sizes and shapes in these brains, ranging from a typical human brain shown here down to, in the lower left-hand corner, this is the Senegal Galago, and that brain is shown to scale. So of course, a huge range of size, very clearly shown here, as well as shape, with the amount of foldedness in particular. And in between, we had 10 other primate species. And I can show a bit more information about them here. The species, they're arranged in order of increasing surface area. So we were really fortunate for this study, which has just been submitted to benefit from a lot of open science initiatives and data sharing initiatives. And we've made all of our pre-processed smooth normalized surfaces and the quantities that we calculated across all of the surfaces available on Zenodo. So you can check that out if this is of interest to any work that you're doing. So with these 12 species, to make it a bit more tractable, we divided them at some natural breakpoints in their surface areas into small, medium, large, and then humans are really our only extra large species here. And I'm going to show here just one brain from each of these groups, but we did find similar patterns in non-human primates. And the rest of the data can be found in our paper that's been submitted. So this is, again, the human data that I showed before, where we see this smooth spectrum from the yellow concave shapes to the blue convex shapes in their cortical thickness. And we see similar patterns in the white-faced saki, the rhesus macaque, and the chimpanzee, and where we see this smooth spectrum of cortical thickness between sulci and gyri. And the inclusion of these 12 primate species allows us to test some of the hypotheses that our earlier computational and experimental models of thickness bifurcation had predicted. And so we can look here at the relationship between the thickness and the thickness ratio. So the average thickness and the thickness ratio. And our stretched polymer experiments back in 2018 had predicted that these would correlate positively. And indeed, we do see that. Another prediction, especially from our computational modeling, was that thickness ratio would increase as folding increases. And again, we do see that. We're using the gyrification index, which is the ratio of the total area, so in all the nooks and crannies, to the exposed surface area. That quantity generally increases as brains increase and indicates basically an increase in folding. And again, we do see that as the gyrification index increases, the thickness ratio increases. So just to wrap up here, the main points that I want to emphasize are that in general, across the brain, convex parts of the cortex are thicker than concave parts, and that this pattern holds across at least a number of non-human primate species. And finally, that this allowed us to test this hypothesis and show that thickness differences increase with both average thickness and with foldedness. So I want to acknowledge the members of my lab and collaborators, particularly the hard work of my graduate student, Agahan Demirci, as well as our funding from NSF right now with a career award. And lastly, if you'd be interested in any more information, as I mentioned, we have the surfaces available on Zenodo already and our code on GitHub. You can find those links on the Command Lab website. You're free to reach out to me by email or Twitter. And we have, again, this paper on humans and out in human brain mapping already. And we've recently submitted the paper on primates. And I'd be happy to discuss more. Thank you.